Good morning, everyone. Welcome to a conversation with Penny Chisholm. I am Andres Cubillos. I was Penny's last grad student. I didn't know what I do to be the last one, but it was 10 years ago that I met her, and I was fascinated by the research that she had been doing in her lab and the unique approach to microbiology. So I decided to join her lab. And as you know, Penny has been the champion of Prochlorococcus. She has been trying to further these microorganisms for the ones that I'm sure everyone know here. What Prochlorococcus is the most abundant, the smallest photosynthetic organism in the ocean. And from the moment of the discovery in the late 1980s to now, she has basically turned this as a model system for cross-scale biology, a way of understanding from the cellular level to the organism level, to populations, to communities, to the whole ecosystem and the global scale. So it's a really important research that she has been doing. And she has received many awards for this. And I know that Penny likes to put Prochlorococcus in the spotlight, not so much her in the spotlight, but I think it's important to mention some of the awards she has received for this work. One of those is the Agassiz Medal from the National Academy of Sciences, the National Medal of Science in 2011 from the White House, the Obama White House. Also, in 2015, she became Institute Professor. The Catalonian government gave her the Ramon Margalef Prize in Ecology. And this year, she received from the Royal Swedish Science Society the Crawford Prize, which is a very prestigious prize for environmental sciences. Besides the scientific endeavors of Penny, she has also received awards because of her outreach activities. You might hear from her in a moment about her children's book. She has written 4 so far, where she teaches both kids and parents, because believe me, like not everybody knows about this, how the sunlight feeds all life on Earth. And those are really beautiful books illustrated by Molly Bang that I can tell that at least my daughter loves. Also, Penny has been able to transcend Prochlorococcus and take it to different levels and to different people, such as President Barack Obama. She even discussed with the Dalai Lama about Prochlorococcus. and she actually warned him about you will talk about that later, so I won't say it. Like, it was a really fun conversation that they had. Even rappers have gone to the lab to get inspiration from Prochlorococcus. A former Wu-Tang Clan member, Jessa, came to the lab and talked to Penny to get inspiration for his new album. That was quite a visit. So throughout these many years, Penny has learned a lot about Prochlorococcus, right? But I think she's also learning from Prochlorococcus. As you know, there are like some ecotypes of Prochlorococcus that can colonize new niches by adapting to different conditions. For example, low light. I think Penny is brave and has done the same. While her house was undergoing renovations, she decided that she would colonize a new niche in the backyard of her house, and decided to colonize this low light, low space shed where she spent over a year while these renovations were completed. So that's some Prochlorococcus courage there for you. But seriously, she has learned from Prochlorococcus and has transmitted that knowledge to us, all her mentees. And I think that the most important thing is the power of diversity, the importance and the power of diversity. Prochlorococcus is a collective of genes and functions that, working together, are resilient, and they accomplish a variety of functions that have global impact. She has led by example in her lab doing the same. bringing people from all over the world, different talents, different ethnicities, orientations. You know, she has made her lab a really welcoming and inclusive place. And she has put the people first, and she has been a really caring mentor for everyone. I remember, I can say that in almost every meeting that I had with her, we started our conversations with how beneficial Tai Chi can be, and then every new mindfulness app that was available for iPhone at that time. So that speaks on how Penny is. Penny has also served as a role model for women in science. She was the first faculty in the Civil and Environmental Engineering department here at MIT. And along with Nancy Hopkins here, they worked together in a committee to expose the inequalities of women in the early 90s here at MIT. And she's gonna talk more about that later. But what is beautiful is that the work that they did resonated not only in other schools here in the nation, but internationally and also outside academia. So she has been also championing women's rights and supporting women in STEM. So with that, I won't take more time from this introduction, and I'm very pleased, and it's an honor for me to introduce Institute Professor Penny Chisholm.
Oh, thank you. That was so wonderful. Well, thank you. You reminded me of all kinds of things that I see. I want to thank Mandana for finally convincing me to do this. Took her several years because it was really interesting for me to kind of go through my life and figure out, you know, how on earth did I end up here? And so here's my life in one page, which I will probably not follow, but and I guess one of the reasons I agreed to do this is for the students out there is to say, you have no idea where you're going to end up. I mean, if you told me when I was in college that someday I'd be an institute professor at MIT, first of all, I wouldn't have known what it was. I barely knew what MIT was. And I just would have said, you're crazy. So, I mean, I pinch myself every day and wonder, you know, how did I end up here? How did this happen? And so I thought I'd just start with the beginning, which is where I grew up, because I think that shapes all of us in some way. I grew up in the Upper Peninsula of Michigan, which most people have not heard of. Michigan has two parts, the mitten and the Upper Peninsula. And I grew up here. Up in this, we were in the largest town in Upper Michigan on Lake Superior, but it only had 25,000 people. And, but it had a university and a prison, and it was a medical center for that area. So it had a lot of things going for it. And it was on the lake, and it was beautiful. And it was just a great place to grow up. I was lucky. My parents were both educated, and we had, you know, we were comfortable. We weren't rich, but I didn't want for anything. And we only went to school for half a day in junior high and high school because that was the baby boom era. There were too many kids and not enough money. And so it wasn't too bad for those that wanted to go on to college, because we just had the basics. They didn't have much of the extracurricular things. But it was great. And so we went to school half a day and skied the other half of the day. And somehow I managed to do all right. And but I had no goals. I mean, I knew I was going to college because my parents said, you're going to college. I was kind of interested in medical technology because I got these brochures at school, and I liked the test tubes and things. And then my parents said, no, that, you know, that's a 2-year degree. You're going to go to college. So so I had to go to college, and my brother had gone to Dartmouth. I had an older brother, a couple of years older. It was a very patriarchal family. A lot of the attention was on him. He was the oldest son of the oldest son of my grandfather, who was a self-made man. He worked his way up from the mines to be a partner in this big mining company. And so there was a lot of pressure on my brother, and no pressure on me at all. Like, it just, whatever. But I had to go to college. So, and he was at Dartmouth, and so because my grandfather had sent all of his sons to Dartmouth. My grandfather didn't have a college education, and he was very into education. So off I went to Skidmore College, which was all women then, because it was near Dartmouth for no other reason. And that was a shock, because I grew up in a small town, in a public school, and suddenly I'm in this women's college, and a lot of women who had been to prep school, and the social life was just nuts. This was, I don't know if anybody has ever seen the movie Animal House. That was my brother's fraternity. So the social life was just totally warped. But I think I got a good education. And that's where I got interested in science. And I did a senior thesis on the manganese budget of a lake. A local lake, and simply because my advisor suggested it, and I thought, well, that sounds cool, and we went and sampled the lake over the course of a year and watched the manganese complex in the sediments and then be released, and measured it inputs and outputs, and looked at the phytoplankton, and suddenly, this lake became a system. It became alive. It was the fact that you could measure this stuff and make sense out of it was just so interesting to me. And so I was kind of hooked. And then I learned that you can publish papers, that you can write this stuff down, and somebody has to believe it because it's in this paper. And it was this huge epiphany, because I think it came from, nobody taking me seriously when I was younger. So, I mean, childhood was just fine, but I think I was always trying to get somebody to say, hey, I'm here. So that was science as a way of knowing. I found, was sort of an outlet, as a way to actually have some authority in the world. And so that was cool. But then I was going to graduate. I didn't know what to do. I certainly didn't want to get a job. And my advisor said, well, you could get a PhD if you wanted. And I had never even thought about that. Nobody in my family was all sort of business oriented, and there were no PhDs or even doctors or lawyers. And so that sounded good. I liked going to school. So I applied to graduate school. And I started at Cornell, where I was working on ecosystem nutrient budgets. And but make a long story short, that was not a good fit for me, and I had a dual career thing going on. I had a boyfriend back in upstate New York. And so after a year, I left and went to State University of New York at Albany. And so I always tell my students, follow your heart, because it'll work out better. Because I ended up in a biology department, and it was when SUNY Albany was, the New York State system was booming. There was all kinds of money pouring into it. Lots of young professors, lots of money, and so that's where I got my PhD. I worked on euglena, so I got into phytoplankton ecology, and I got interested in how they respond to a light-dark cycle. In fact, I was astounded when I learned that people grew them in continuous light, phytoplankton in culture, and I thought, why don't they explode? I mean, because they're not used to that. So my thesis was about how they respond to the light-dark cycle, and my office at the university was filled with people working on circadian rhythms, people working on neurospora, aplesia, and rhythms in neuronal activity, and all these different areas. And so it was a real hybrid. So I was the only ecologist, but it was a hybrid environment. It was a biology department. I mean, sorry, this is a biology department. But it was an everything biology department. So ecology and cell biology, all of that was under one roof. And when you took your general exams, you had to have 3 out of 4 of these different disciplines. So it was exciting for me. And also, I took a genetics course then, which blew my socks off. And this would be 1972 or something. And it was a neurospora geneticist, and he was working on circadian rhythms, and it was just getting very tractable with genetics, and I thought, wow, this is really cool. But somehow the lure of the natural world, even though ecologists were kind of like second-class citizens in that department, the lure of the natural world stuck with me. And I went to Alaska for a summer and worked on zooplankton in ponds, and so anyway, so then, of course, then I had to do something because I got my PhD. So I went to Scripps Oceanographic Institute as a postdoc. And that's when I got into big oceanography. I realized where the money was, the funding was. Limnology is kind of not a lot of money. But oceanography, the Navy was investing a lot of money then. And so that was exciting. And this was the 70s in California. It was totally wild. I will not tell you stories. They would shock that when I think about what's not tolerated now and what went on then. It was just, my office was right on the beach, and you just put your bathing suit on and walk down to the beach in the middle of the day. And it was pretty wild. But I had a really great mentor there. Did some interesting work on, I was still interested in cells and how they divide, and I was using silicon uptake and diatoms. Diatoms are phytoplankton that have a silicon frustule that they have to reproduce when they divide, and so we used germanium as a tracer for silicon, and we were trying to use that to measure their growth rates in situ, right off the Scripps Pier. And couldn't get them to take up any germanium. And finally, my colleague and I looked at each other and said, maybe there are no diatoms out there at this time of year. So we added some, and voila, there it was. So that was another chapter. So then I had to get a job. And at Scripps, they only let you be a postdoc for 2 years. So I finally had to get a real job. Applied lots of different places. Long story short, I actually gave a talk at a chemistry symposium. I can't remember for the life of me why I was there, why I got invited to that. But Francois Morel, who was a chemist in the civil engineering department at the time, was there, and he heard the talk, and he said, "We have a position, you should apply." And I thought, why would I apply to a civil engineering department at MIT? But I was applying, and they wanted a biologist to supply parameters for water quality models. And anyway, long story short, I interviewed at different places, and I got an offer here, and couldn't decide what to do, because one of the other offers was an oceanography department. I would have fit right in, felt really comfortable. And I had a mentor at Scripps who said, "Penny," he was a Harvard guy, he said, "Penny, you don't turn down MIT." I said, okay. So I came, and that was quite a shock, because I got this letter from the department saying, by the unanimous vote of the faculty of the civil engineering department, you have been selected, blah, 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 blah. I thought, well, that's cool, they want me. But of course, you get here, and I was like, who are you? There was somebody in the lab where I was promised that I had to figure out how to get out. And we ended up having to build a lab from scratch. When I look back, it was just crazy. I mean, physically, my graduate students built the lab. They put in the benches, they put in the countertop, and not mine, but the group, Francois Morel's graduate students too. I only had one at that point. So there I was, trying to figure out what I was doing in a civil and environmental engineering department, madly teaching ecology, which I felt, really excited about, and was sort of developed this class from from scratch, which still exists, I'm very excited to say, and is still the only introductory ecology class at MIT. And I always was interested in watching the numbers of students would go up and down depending on whether the environment was in or not. And it peaked at 80 one year, and I don't know how many there are now, but it's definitely shrunk. So in terms of my research, when I got here, I continued to be interested in phytoplankton and how they interact with the environment, and not just the light-dark cycle, but the also pulses of nutrients, and we did a lot of chemostat experiments, and I got interested in cell cycle. Cell kinetics were kind of big then for the old timers here, might remember that, and yeast, and so I got interested in how individual cells, the variation between individual cells in a population. And that's when I met Bob Horvitz, and because he had a video system that measured development, right, in your worms. Anyway, a time-lapse video system. And so he showed us how to build this thing, and we looked at the growth of phytoplankton on petri dishes. There were these wonderful videos, just watching them, and then monitored cell-to-cell variation and, you know, daughter cells, and long story, but some papers came out of that. Also, Bob and Graham were stewards of the joint program here in the biology department at that time, which was strongly supported by Salvador Luria, I guess. And they were young assistant professors with me too, so they did what they were told. And they were very good to the joint program students. Joint program with Woods Hole, for those of you that don't know what I'm talking about. But a lot of my students were drawn from that program. So let's see. Okay, so fast forward. So worked on eukaryotic phytoplankton, several different species. It was interesting, basically doing cell biology. But so we also needed, if I was going to do cell biology, we needed to do flow cytometry to look at them progressing through their cell cycle using DNA stains. And we didn't, probably there was a flow cytometer, I'm sure there was a flow cytometer over here, but this was like a foreign world to me then, you know, so we thought, well, we can't afford one of those big things. And so I had a very talented postdoc, Rob Olson, and a very talented research associate, Sheila Frankel, neither of whom were electronics people, but they were just smart people. And they built the flow cytometer in the lab, a microscope-based one. And we did cell cycle analysis on these eukaryotic cells. And then we realized, well, this is really cool. You can measure fluorescence of individual cells and light scatter of individual cells and see what their size is, and you can measure their DNA. And wouldn't it be cool if you could do this at sea? You have all this diversity of phytoplankton. And what would happen if you just put seawater through a flow cytometer? Maybe you could resolve all these different things. And so long story short, I managed to get funding to get a big rig, real laser-based system and everything. And Rob managed to make it work on a ship. That started our journey. We started by studying this tiny cyanobacteria called Synechococcus, which is a relative of the Prochlorococcus that Andres talked about. Because I had been inspired by John Waterbury, he's retired now, a scientist at Woods Hole, and he had taken weekly samples off the coast of Woods Hole of this Synechococcus, which is just a tiny little red fluorescing unicellular cyanobacterium. And so he had this time course of how its numbers changed with the season, and he was also studying phage infection. And I'd always been frustrated that the species that we were studying in the lab, we couldn't go out to sea and find them because they're diatoms and coccolithophores, and who knows when they're going to bloom or where they're going to be when, or you just can't count on going on a cruise and finding them. And I always wanted something that I could study in the lab and at sea. And when I saw that you could see these cyanobacteria easily, and they're everywhere, the Synechococcus were everywhere, I thought, well, this would be really cool. And you could see them beautifully with a flow cytometer. So we set out to study them, but we were surprised when we saw something even smaller than them. Rob first noticed them in the flow cytometry signatures that were tiny little red fluorescing cells that were smaller than anything that was supposed to be alive with chlorophyll. And that turned out to be Prochlorococcus. So when we found those, I said to myself, this is it. I'm going to do this for the rest of my life. There's something about them spoke to me, their tininess, their abundance. And even though they're not off the waters of Woods Hole, which we still don't understand why, you have to go out to the Gulf Stream to find them, because they'll grow in Woods Hole seawater if you filter it and bring it back to the lab. So I think it's an ecological, I think they're eaten away. I think they're there, but they're so rare that you can't see them. But that's another story. Anyway, so that became the Prochlorococcus journey. And at first we just studied their physiology. Growth rate as a function of light intensity. Young graduate student, Brian Palanik, finally got them into culture. And so we studied them for about 3 or 4 years before we could we studied them at sea, just by manipulating the seawater and stuff to make sure they were really alive and doing things. And so once they were in culture, and then one of my former graduate students isolated another one from the Mediterranean Sea, and we realized that they were very different in their physiology. So isolated some more, they were different light optima, light intensities that would kill one, the other one would thrive. I mean, they're really dramatically different physiologically. So that was interesting. And then eventually we were able to sequence their ribosomal RNA and show that they're very closely related, even though they have this very diverse physiology. And in 2003, we were lucky enough to be among the first microbial genomes sequenced. I think Prochlorococcus was the second one. And I convinced the JGI, the Joint Genome Institute, that sequencing 2 would be really powerful. And since the genomes were small, maybe we could get a twofer. And they did, and that was the beginning for us of really opening up the black box. Or me going down the tunnel of reductionism, I say, which I'm constantly fighting. It's like, so the genomes were dramatically different and had these signatures that we could map onto their ecological niche. And so in my mind, that's when we started doing ecological genomics. And luckily, at the same time, that in 2004, Craig Venter, all of his human genome sequencing machines were idle, because that was done. And so he set sail with his yacht and went around the oceans, collected seawater, and poured it through his sequencing machines and got, sequenced all the microbes. But luckily, Prochlorococcus was highly represented in all of those samples. So we had the genomes from the lab, and then through all these ocean samples, we could recruit the Prochlorococcus genes, get them away from all the other stuff, and look at the distribution of the genes in the global ocean. And you could see patterns. You could see, we know, for example, the Atlantic Ocean is heavily phosphorus limited versus the Pacific Ocean. And you can see that in the genomes. The Prochlorococcus in the Atlantic have many more phosphorus transporters and different ways of accessing phosphorus. You can see that in iron-limited regions versus non-iron-limited regions. You can see siderophore transporters and things like that. So it was just incredibly exciting and that's when I realized that the cells have stories to tell us if we just listen to them. And I've always just let Prochlorococcus be our guide. Well, Prochlorococcus and all of the incredible graduate students and postdocs I've had in my lab. So I've never assigned projects to anybody. I just say, okay, here's what Prochlorococcus has to say, you know, which of these stories do you want to listen to, and what do you want to follow? So it's just been incredibly easy to just follow what the cells are telling us. Well, it's not always easy. I mean, sometimes I feel like they're screaming at us. You know, why can't you see this? You know, there are these. In fact, what Andres worked on, these prochlorocins are secondary metabolites that Prochlorococcus has this tiny genome, and it makes, I don't know, 10 times more of these than your average bacterium that they're antibiotics or signaling compounds. In Prochlorococcus and Synechococcus, they just make a ton of these, and we have no idea why. And that, you know, one of my colleagues always says, you know, Penny, you know, why are you so obsessed with these prochlorocins? And I said, because they're trying to tell us something. And Andres made some good progress on that. Okay, so we have more and more genomes, and we realized that although each cell has about 2,000 genes on average, but they only share about half of those. And then the rest are sort of these niche specific genes. And the pangenome of the whole collective, the billion cells in the ocean, is about 80,000 genes we've extrapolated. So this is a superorganism. I think of it. I think of it as one. Sometimes we call it a collective. Sometimes we call it a federation. But it functions as a superorganism, as do all the other bacteria out there too. But this one we've got a pretty good handle on, and it's taught us a lot about the oceans. I had a graduate student, or a postdoc, excuse me, that discovered that Prochlorococcus buds off these little vesicles, tiny lipid-bound vesicles, like other bacteria do, but they were not known in ocean ecosystems. And he was able to show that they contain DNA and RNA and prochlorocins now. And because we had these in Prochlorococcus, we could then go out in the ocean, collect the whole vesicle fraction, sequence the DNA in those vesicles, and tell you what bacteria they came from. And so this is a whole new feature of ocean ecosystems that we know essentially nothing about. We don't really know their functions, but people are working. Steve Biller, this is his work. He's spreading the techniques and stuff, so other people are working on it. The other thing about this system that we learned, we spent years trying to get Prochlorococcus azenic. We isolate it from the oceans, but it brings along its bacterial friends, and it does not like to grow without them. So we spent years, and we now know some of the mechanisms, some of why they complement each other. So we spent years getting them azenic, because how could we be, respectable microbiologists if we're working with contaminated cultures? So we skirted around all the questions that you can't answer with azenic, that you can't approach with azenic cultures, and so you can look at light adaptation and things, without worrying too much about the bacteria. But but we ran out of questions that we could answer, so we finally got them azenic, and then we realized, they're not happy. And that the phenotypes we measure in pure culture are not the phenotypes that exist in the wild. And so then we started adding systematically the bacteria that they dragged along. In fact, Sean, who's here, who's a postdoc, is actually looking at all of our cultures and what communities they dragged along with them over the last 20 years. And then we add them back systematically to see what role the different cells accompanying Prochlorococcus play. And so the more I think about this, think of how much of biology, our understanding of life, is built on organisms in isolation. So I've decided all of biology is wrong. And that's a little strong. But I think it's profound, and the more we learn about how different the behavior at all levels, the molecular gene expression, proteins at all levels, when you add one more microbe to the system, you get a very different result for many important phenotypes. So, so with that, you know, I have a favorite quote that is one of the things that kept me in ecology. It's a famous ecologist. His name is Slobodkin. And it's what keeps me from really going. I've noticed over the years that our questions have become more tractable now that we have molecular biology, xenic cultures, genomics, and that we keep going down the tunnel of reductionism because the next question is obvious and the way to approach it is obvious. And I keep fighting that because so this quote from Slobodkin is what kept me, he was around when I was a graduate student. He said, "Ecology is intractable in contrast with physics. Physics is free to decide what is or is not within its own domain." And he says, for example, meteorology is not physics, even though it is, because it's not tractable, right? Sciences whose domains are defined by their capacities are tractable in a way that is impossible for sciences defined by a domain of subject matter. And so I think the power of molecular biology has become defined almost as molecular biology now, I think, and because it is so powerful and so tractable. And now I'm experiencing this. See, I read that quote when I was a youngster. And now I'm experiencing this in my own work in real time, the power of reductionism and tractability. And I keep fighting it because now when I go to conference, I'm part of a big oceanographic collaboration that's funded by the Simons Foundation, the Simons Collaboration on Ocean Processes and Ecology. And it's big oceanography. You know, everybody goes to sea and measures all this stuff, and they tolerate me, even though we're doing all this, trying to figure out what prochloricins do. But every time I go to those meetings, it pulls me back. It makes me realize that yes, these, you know, there's all these uncertainties in these questions, and you know, they're intractable in so many ways, and yet that's life. It's the whole system. It's the Earth, you know, and that's the living system. So I could go on and on forever about that, but before I close, I wanted to, because people always want to ask about my experience as a woman in science, so I wanted to say a few things, and then I want people to ask me questions. So I was always growing up, my parents' friends all had sons. So I grew up, and I had a brother, and I just grew up among boys, and I was kind of a tomboy. And when I got into oceanography and everything, you know, I kind of liked being a woman among men. I mean, and I was young, I was single, you know, and it was party time. So it was fun. And I didn't think about being isolated. I don't know why, but it was fun. And then I came to MIT, and again, you know, I was surrounded by men, but it didn't feel that strange. But then there was a colleague of mine, man, who lived in Newton. They had a carpool of faculty and research faculty in Newton that commuted, and they asked me to join, and I lived in Newton then. They asked me to join the carpool. And I said, well, you guys are all married and have kids and 8 to 5. And I said, I don't go home at 5. I go out and party and, you know, whatever. So they said, okay, but come for breakfast on Friday. We always have breakfast at the Hyatt on Fridays. So I said, okay. And so off I went on Fridays with these guys. And I realized that they had, most of them, gone to MIT all the way through. They were lifers. You know, back in those days, a lot of the faculty that were hired from within. And, excuse me, and And I realized talking to them that they were experiencing this world in an entirely different way than I was. And I don't have time to go into the details of it, but it just seemed so easy for them. And I'm like holding on like this, just trying to get through the next day, And they're volunteering to be on these committees on commencement and stuff. And I'm thinking, why on earth would you take on something you don't have to do? And they said, well, it's fun. And I said, fun, fun. And then one of them, this is really fast forward, but I try to remember the pivotal times, and most of them it was after I got tenure. I joke with my colleagues, my feminist needle went bong, and I don't think that's coincidental. But one of my colleagues gave me this book by Carol Gilligan. It's called In a Different Voice, and she was a researcher at Harvard. I don't know if she's still there, but it was just about the differences between men and women and childhood development, and it wasn't about science or anything. And it really spoke to me. And also I read a book by Evelyn Fox Keller on gender in science, and I sat in on her seminar here, and I went, oh, wow, I've really been missing a lot of stuff here. And that was just about the genderization of science, and not about equity or any of that stuff. And then he gave me a book by Catherine McKinnon, who was a radical feminist at Cornell at the time. So I woke up and I realized that that I was just experiencing a different world. And the way I describe it is that there's a playbook for this whole enterprise we're involved in, and it was written by men. And so I think I felt as a woman, you're just constantly trying to figure out what the game is and what the playbook is, and did I do that right? Did I answer that right? Should I do this? Should I do that? Should I? And it was just started to become obvious to me. And then I came up for a promotion. Well, I didn't come up for a promotion. I was supposed to come up for a promotion to full professor. And this is something I don't talk about that much, but now that I'm an institute professor, I can. And it was just the time, normal time. There's no real time to be promoted, but and the department head at the time was a new department head. He had a lot of tough tenure cases on his plate, and I'm sure you can relate to this, the department heads. And I was impossible to describe. What was this phytoplankton ecologist doing in his department anyway? He didn't say that, but it was, so my promotion cases were not easy because I was so weird to be here in the civil engineering department. But I really felt, so he just wanted to delay it a year. He said, everything's fine, but it'll be a lot easier next year, blah, 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 blah. And I just felt, no, there's money involved here. And I think I deserve to be promoted now. So I had a wise person then that I found who knew a lot about MIT, who I could go to at times like this. And he said, there's an associate dean over there that you should probably take this to. And so I went to the department head and I asked him, I said, can I go over your head? And he said, sure. And so I went and talked to the dean, associate dean, and I said, I really feel I should be promoted. And I wrote this long thing. The issue was leadership. And as I said, I'm just like hanging on here. Lead. First of all, there's nothing to lead at that point. But anyway, and so I wrote this memo about leadership and about leadership by example. I said lead. I said just surviving here this long is leading, and I'm exaggerating. But anyway, it made me think about different forms of leadership and put it down on paper. And he said, I'll support this. He said, if it doesn't go, but you should know if it doesn't fly, you have to skip a year. In other words, you want to play it safe or do whatever. And I said, yeah, let's go for it. And the department head was fine with it, and so it went fine, and I got promoted. And that was my first lesson. And if you don't ask, you don't get. And it's not this system out to get you or anything. I mean, he was a good guy. He didn't mean he was supportive of me, but he just thought it'd be easier if you waited a year. And of course, after that, I became the director of the joint program. I became the associate chair of the faculty. I became who knows what. So I think again, it's complicated. And then, okay. Nancy Hopkins came along, and that was the next chapter in Women in Science. And by that time, I was a full professor for years, and things were going great. And the senior women in the School of Science, led by Nancy, got together and realized that nothing was changing in terms of numbers of women and on the faculty and equity in salaries. Several times over my career here, I've gotten a bump in salary, unexplained, which of course showed me that I was underpaid and somebody noticed. So that was something we could spend a lot of time talking about. But the final thing I wanted to talk about was teaching. As I said, I taught introductory ecology for years and years and years. And then I remember you were involved in that. You probably don't remember this, Bob, but I was on sabbatical when you decided to make biology an institute core. And I emailed Bob, who was, I don't know why you, but because I knew you well. I said, if they're going to make biology required, there has to be some ecology in it. I was on sabbatical and thinking this would go nowhere. But Boris Magasanik actually invited me to teach part of the flavor that he was teaching. So we managed to slip 10 lectures of ecology in the institute core, which was fun. But it was through teaching that I learned that photosynthesis is really not in the curriculum. And it's ridiculous. The whole world, nothing would exist without it. So I got obsessed with photosynthesis. That all of life depends on photosynthesis, and there's this study done by the Harvard Smithsonian actually interviewing MIT and Harvard graduates on graduation day, giving them a log and asking them where the weight of that log comes from, and they don't know. They say water, and then they say, what if I told you it came from the air? And an MIT student, who, poor thing, luckily nobody can find this except me, said, well, I'd say that's probably impossible because it would have to be compressed, and anyway, so, and every year I would ask the students in 7014, like a couple of weeks after I gave the lecture, where the mass of their body came from, and they couldn't answer. It's just something so hard for us to embrace, that we come from the air through plants, period. So I wrote these children's books, and which as Andre said, I wrote really for adults. So this one's about photosynthesis on land. This one's about phytoplankton, photosynthesis in the ocean. This is about buried sunlight, fossil fuels, ancient photosynthesis. And this one's about the global water cycle that makes it all happen. So I'm going to give these to, so who has a child or grandchild between, that's 6 years old? 5? Okay. Now, there was, no, I had this algorithm. It's not working. Okay, I'll give him to Monty. He's in the front row. But if you want some, if somebody else wants some, come. I have a stash, so you can come. Or you can get them on Amazon. Yeah. We have a website, thesunlightseries.com. So finally, just the last word. I was listening to Wait, Wait, Don't Tell Me the other day, and Henry Winkler, the Fonz, the youngsters don't know, but probably who that is. But anyway, they asked him what his key to success was, and he said perseverance and gratitude. And I said, I could use that. And I would add something called luck that actually goes first. Luck, perseverance, and gratitude is huge. I mean, anytime I felt down about anything, I had to think of everything wonderful that I have had at MIT. The students have been wonderful, the whole philosophy of MIT, the meritocracy, even though sometimes it doesn't work. I just would not be here if it weren't for MIT. So I thank you all for coming. I'm sorry, I went on so long. But there's time for a few questions.
I'm tempted to talk about the latter stuff, but I had a burning scientific question.
Sure.
So for these microbes that are unhappy when they're separated, do you see evidence of horizontal gene transfer between them?
Not.
Have you looked for such?
Some people are trying to look at that. Definitely trying to look at that.
Yeah.
But luckily, some of my people are back there. I don't think we've seen anything yet.
There is with Prochlorococcus, actually.
What?
With Prochlorococcus, yeah. I don't know if in cultures,
But not between. But not between the ones that live together in the culture.
I'm just trying to understand, is it all small molecules that are going back and forth?
You're talking about in the culture.
DNA that's also an element.
You're talking in the cultures, not in the
culture
world.
Or
In the world, definitely. In the world, it's a giant sea of genes. I think of the ocean is just a giant sea of genes. I think more.
I wonder how much of that gene transfer is just fortuitous and how much of it is actually knitting these communities together somehow. That's sort of the question.
I think there's a lot of trial and error. I mean, obviously,
Yeah.
These vesicles are floating around with DNA in them and fusing, and we also now are looking at some little integrative elements in Prochlorococcus that pop out and replicate themselves, and are involved in phage defense, and it's a jungle for sure. Yeah, there's a lot going on. Any other burning questions?
I don't have a burning question, Penny, but having taught with you over those decades and seeing it with the flux, I just wonder whether you could share your technique you used to try when we had very big lecture halls and trying to get the students closer so they could hear your magic of the photosynthesis. You had a little algorithm there that did work.
The bribe?
Yeah.
I used to show a picture of the seating chart of the classroom, these huge classroom, and they all sit in the back, so you feel like you're teaching nobody. And I really thought they would think this was totally bogus, but I had a random, well, it wasn't random, but sort of random number generator in the beginning of class that would circle in on a certain seat, and that student that was in that seat, there was a no prize zone in the back. And that was if they sat in the prize zone, they were eligible. They were eligible for a prize, and they were always the same students, just sat in the no prize zone. They didn't care, they're working on their whatever. But the ones in the front, so I'd give out free books and food or all the swag in my office. Those backpacks you get at meetings and
Giant clam.
yeah. One time I had a piece of a clam from a hydrothermal vent ecosystem, which was really precious, and I had a piece of one, and the student that got it was going on to graduate school in that field, and she was absolutely beside herself. I mean, that was really so. Yeah, it worked. It got old after a while, but it worked with some students. Any other questions? Answers? Is all biology wrong? I hope not. But okay, well, thank you all for coming.